These beating heart cells were created using embryonic stem cells. Soon, cells like these could be grown using stem cells produced from fetal skin cells, taken from the amniotic fluid of a pregnant woman. Currently, stem cells can be produced from adult skin cells, but researchers at New York's Mount Sinai School of Medicine have found that fetal skin cells floating in amniotic fluid are far more efficient than their adult counterparts in producing valuable stem cells. Lead researcher Dr. Katalin Polgar says the news is exciting because once the fetal skin cells are reprogrammed to become stem cells, they could then be directed to become any one of the 200 cell types in our bodies.
Those cells have a very young epigenetic background. It means that they are amazingly fresh and new and robust. So when we work with them, we can see that they are much faster, much efficient when we try to induce them or so-called reprogram them to be a stem cell.
Skin cells are shed by the fetus as it grows and are easily extracted from amniotic fluid drawn from a pregnant woman for routine tests. The resultant stem cell line developed from stem cells would be unique to that child and could potentially repair problems while the fetus is still in the womb and later in its life.
We turn back the clock of a final cell type in the amniotic fluid, back, or should I say reverse them to their first starting point, which is a stem cell. And why it's so important? Because we are able to, number one, try to produce a cell bank for possibly use for the fetus. And also, we don't need in this time to destroy a human embryo, which is very important.
The science is still in its research stage, and Dr. Polgar hopes that this powerful tool will soon be readily available to help cure diseases. Tara Cleary, Reuters.